All right. Hi, my name is Subaita, and I'm a second-year life science student, and I'm currently working at the Moving Lab. And today I'll be presenting the validation of an image analysis algorithm for 3D hydrogel culture platform glans. So it's now recognized that the tumor microenvironment plays a significant role in tumor development, metastasis, and progression. As you can see in this figure, there are cancer cells in a tumor and its surrounding microenvironment indicated in the blue. It's next to a blood vessel which can help it grow. Cancer-associated fibroblasts, known as CAFs, immune cells, and endothelial cells are all found in the tumor microenvironment, and it can secrete growth factors which contribute to drug resistance, and it should be accounted for in the drug discovery pipeline for new cancer drugs. Currently, there is a lack of relevant tumor models in the drug discovery pipeline. With 2D cell line assays, they're easily scalable, convenient, accessible, and low cost. However, they're less biologically representative and produce non-predictive results in drug screening. In vivo mouse models produce better representations of human systems, but they're mouse cells. There are long assays, high cost, labor-intensive workflow, and there's limited control. An ideal tumor model would be a 3D in vitro tumor model, which can simulate a 3D architecture, cell-to-ECM interaction, cell-to-cell interaction, modulation over a relatively short time, and better drug prediction. That's where glans comes in, and the Moving Lab has previously published a 24-well plate version, but we're currently expanding the 96-well plate, which is the one I'm working with. So glans sells for gels for live analysis of compartmentalized environments. So first you have a seeding stage where you have your PDMS slab with your inlet and your outlet holes, and you also have your chip with 96 channels. You then have your cells, which would be embedded in a gel, and you'd seed it through your inlet and your outlet holes. You then let your chip incubate for about 37 degrees for 30 minutes. So the next step is assembly, where you would remove your slab and you would have your chip sandwiched in between a sticker and a 96 well plate. This would essentially create 96 tumor tissues, each with their own microenvironment, where you could add drugs into the plate and then look at results. So here's a top-down view of what glans would look like with its channels seeded. And as you can see, there are artifacts, which are areas of higher or low concentration of cells. So there could be areas where there are no cells indicated by this yellow arrow here, or there could be areas of high density cells. This slows down the time it takes to acquire meaningful data from each channel. So when we're doing image analysis, we want to look for our mean gray value, which can be used as a surrogate for our density, since it has a linear relationship. So currently we do our image analysis in Fiji, and we'd have to manually move around our region of interest to exclude any artifacts or holes. This would be very time consuming, as we'd have to do this for around 96 images, and we'd have different reps as well, and we'd have different days, so it would be very time consuming. Our objective is to develop a Python algorithm that can detect regions of interest, known as ROI, and eliminate any areas of high or low densities in glans images, known as artifacts. Our goal is to validate our algorithm by comparing outputs, hands-on time, variation of plates between current method and the algorithm, as well as demonstrating the usage of a kernel in selecting areas of low variance. For our algorithm overview, we have our input, where we input one batch of 96 images of the plate. So each image would look somewhat like so, where we have our channel boundary and we'd have our artifacts. Next, our algorithm would recognize areas of high or low density of cells for each channel. Finally, you have our output, where we'd have a CSV file with their mean gray value, as well as the ROI for each corresponding channel. This ROI would be within the channel boundary, and it would exclude any artifacts. So our algorithm would map the pixel intensities of each row and crop the image at the first peaks of the graph, which would indicate a channel boundary. So you can see that over here, that's where the cutoff is for where the channel boundary would be, which is around 800 and around 300. And that corresponds to the peaks in this graph, where our y-axis would have our sum of pixel intensity at each row, and our x-axis would have the coordinate of each row. The same would be done for our columns as well, but since the algorithm can't really identify the difference between a row and a column, the image would get flipped. So these green areas here would correspond to the green areas in this image here. So with our cropped image, we'd apply a window of variance, which would be around 2/3 the size of the cropped image. So if we were to split this image into 3 pieces, essentially 2/3 of that would be our window, and we slide that around to look for areas where there's least variance or where pixel intensities aren't as varying. So in this graph, you can see this area here doesn't have too much of a variance of pixels, whereas if we move this over to the right, we'd have a very big difference of pixel intensities. This would likely be an area of an artifact where, again, like I mentioned, there would be an area of low or high densities of cells, which indicate to wilder variance of pixel intensities. This final region would be our ROI. Next, we'd have our kerneling step. So an averaging kernel gets applied onto our ROI and serves as a step to ensure that outliers are analyzed. The final value extracted is the median of all the gray values here in the kerneled image. So you might be asking what a kernel is, and essentially it takes a window that is smaller than the image size itself. So in this case, it would be 3 by 3. It would take all the values within that kernel and divide it by the number of elements in the kernel. So in our case, we have a 3 by 3 kernel, which would mean 9 elements. We then replace our center value in the kernel with a new value, and that would become our new image, which would then take the median of all those gray values, and that would become our mean gray value. Moving on to our algorithm validation, we looked at the hands-on time between different reps and the minutes it takes. So in our manual case, we found that it takes around 10 minutes for rep 1, but for rep 2 and rep 3, it takes around 5 minutes. But for our algorithm, however, it consistently takes around 1 to 2 minutes. It also produces a lower standard deviation than the manual. We also looked at the variation between 3 glance plates, as manual is in the red and the algorithm would be in the black. And we found that there is little variance, as there is a maximum difference of 1 compared to our manual mean gray values, which would serve as our baseline. We also looked at different regions of interest size. So as I mentioned previously, our algorithm kind of arbitrarily draws our 2/3 window size, and that would become our ROI. In our manual, however, we have a constant size of 300 by 600, and we wanted to see if we have a constant size in our algorithm, if that would produce any skewed results. We found that in rep 1 and rep 2, our variable ROI would provide the closest mean gray values to our manual method. However, a constant value can be used. We also looked at the varying dimensions of an averaging kernel. So the example I showed was a 3 by 3 kernel, but we'd like something a lot bigger than that to get more of the image. So we looked at 100 by 100, 400 by 400, and 200 by 600. We couldn't go any bigger than that because anything bigger would be the size of the image, and we can't have a kernel bigger than the image itself because it always has to be a bit smaller to ensure that it can go through each area of the image. We found that 200 by 600 would be the most optimal, as it gives mean gray values that's closest to the manual values in rep 1 and rep 3. We also looked at the presence and absence of an averaging kernel. We wanted to see if our algorithm can perform well with just our window of variance step. And we found that the mean gray values are still quite similar to the manual mean gray values for both cases. However, we need that kernel, as we found that when we ran our algorithm without our kernel, we still had holes or artifacts in our final ROIs, which can skew the final images. The user would have to go back and find those images and either delete it or exclude it from their studies, which would make our algorithm more time consuming. In summary, our algorithm has been proven to complement glance. Kerning provides a great advantage to glance channels, and on average, our algorithm took around 5 to 10 minutes less than the manual. There is little to no difference of mean gray values when compared to our baseline values. Our future step is to allow the analysis of multiple folders and flagging poorly seeded channels. As I talked about before, we'd often have multiple reps per day, and we'd likely want to see the tumor growth over a period of days, so we'd want to analyze multiple folders at once. We'd also want to modify the algorithm for the presence of multiple fluorescent channels. So in this image, we have our CAFs in green, so we'd likely want to modify our algorithm so that it can still produce mean gray values with different images. We'd also want to access 3D information from wild fluorescent image analysis. In conclusion, our algorithm can be used as a tool to research mechanisms of tumor microenvironment-induced regrowth and identify new drug compounds. This could result in a new drug that could not only treat cancer, but prevent cancer regrowth. Lastly, I'd like to thank Dr. Allison McGuigan, Mila Wu, and the rest of the McGuigan Lab. Thank you for listening, and I'm open to questions.
Okay, thank you. If the judges would like to ask a question, could you please unmute yourself now?
Great presentation, very nice flow. I just had a question. So obviously with different experimental conditions, you could be dealing with cells in various states and also spread around the plate. What are other potential issues you might encounter with this algorithm and artifacts that could have come across that you'd have to consider? Excuse me, I don't know too much about this space, so maybe it's a naive question.
So as I mentioned, we only found that there are really just 2 or 3 types of artifacts. So we have like these holes that can occur in the middle of the image, or we could have areas of high concentration. So our algorithm, we also have areas where we might have the cells going away from the channel boundary itself. So we tested our algorithm with all types of artifacts, not just one. And so we found that it's quite robust when it comes to different types of images or artifacts.
Okay, great, thank you.
Hey, Subaita, great presentation.
Hey.
Love doing research like this over the summer. Super cool. I was wondering, why are you choosing mean gray value as a metric? Is there another metric that you would use, or what's kind of just the justification for that?
So we looked at we kind of looked at different cell densities, and we found that there is a linear relationship between mean gray values, so we could use it as a surrogate. So that's kind of our justification for that. But we could look at different metrics to measure tumor growth over time. But we picked mean gray values as we found that it's the closest towards just using the cell densities.